Hi, everyone. I'm Jill Feldman. I'm one of the co-founders of the EGFR Resistors, and we just wrapped up our 7th annual EGFR Resistors Research Summit, which was held in Chicago, November 6th through the 8th. This year, we brought together 15 early career investigators from across the United States who are all doing work in EGFR positive lung cancer. They presented their research and received thoughtful feedback from 5 outstanding mentor judges who are all leaders in lung cancer, chaired by Dr. Zosha Piotrowska. But beyond the formal presentations, the real magic of the summit happens in the informal moments through collaborative relationships that are built, hallway conversations, small group discussions, and time together over meals. I had the privilege to sit with many of these early career investigators and hear about their innovative work and see firsthand how passionate they are about advancing research. It is incredibly energizing and exciting to know that they are the future of lung cancer research. And this year we did something a little different as well. We asked the early career investigators to submit questions in advance of a Q&A that we would have with our mentor judges. And we received a lot of questions. So the Q&A panel with our mentor judges was a huge success. It led to these honest and practical discussions about careers and research directions and how to partner with patient advocates and in a meaningful way to drive the future of research and care. It was really interesting for me too to see this organic conversation happen in real time. So I think we'll be doing that again. And then with the formal presentations, we opened the summit with actually a virtual talk from Dr. Alvaro Guimarães, who was formerly part of Dr. John Haymack's lab at MD Anderson and currently is back in his home country of Brazil. But he shared research that he did using a synthetic cell line derived from EGFR Resistors co-founder Ivy Elkins' raw NGS data to study a rare BRAF TRIM24 fusion, which is the mechanism that drove her cancer progression. So Dr. Guimarães' work his team helped characterize this TRIM24 BRAF fusion and characterize the resistance mechanism. And they also then went on to explore different targeted agents, including one that is not named yet, NST628. It is a pan-RAF MEK in molecular glue, they call it. And then also the MEK inhibitor, trametinib, and looked at both of those as potential strategies to overcome acquired osimertinib resistance mediated by this BRAF TRIM24 fusion. And, what they found actually is important information for treating future patients. Sadly, Ivy didn't live long enough to benefit from this research herself, but her impact continues and her voice helped shape research and now her cells are helping to drive progress in research for other people. And, Ivy also helped shape this summit from the very beginning, in her resilience, her sense of humor, and ability to offer hope in even the hardest moments will continue to guide us as we move forward. With that, I'd like to turn it over to Dr. Zosha Piotrowska to briefly summarize the research presented at the summit and introduce our award recipients.
Yeah, thanks, Jill. I think it's, as you said, it's really such a special event, this summit. And I think starting it off with Alvaro's talk was such a special way to both sort of, honor Ivy's contributions to the summit and to the broader lung cancer community, but also to show these young investigators the power of, what we can achieve working together. I think Alvaro's project really sort of came from discussions between Ivy and other patient advocates and Dr. Hamak and members of his lab. And it really shows, how important these sort of interactions are and how much we can accomplish all together. So I thought it was such a special way to start off the meeting. And just in general, to say as someone who's participated in this meeting from its inception, I think it's such a special event because as you said, it really gives young investigators an opportunity to meet faculty mentors. It gives us faculty members the opportunities to meet these rising stars of lung cancer. And I think, it's really also such a diverse group of young investigators. Each year we get everyone from basic scientists to, oncology fellows, to radiation oncologists or surgeons, sometimes even to medical students or residents. And it's really cool to see sort of the breadth of projects that they bring and most of all the enthusiasm that they bring, and I think a huge goal of the summit is not only to sort of have that formal mentorship, but is to establish, to kind of build this network of investigators and researchers in the space together with faculty mentors, together with the patient advocacy community, so that we all work together sort of with the same goal in mind. So I love the summit, and it's fun to look back on it each year and see, how much we've accomplished from year to year. So with that, we thought it might be nice to sort of highlight the research that was presented. There was 15 investigators, as you just heard, so we won't go through each project in detail, but sort of touch on them and chat about the ones that we thought were impactful or particularly sort of interesting or timely. So, I think all of the work was really great. The other thing I'll say before we talk about specific abstracts is this year in particular, we had public speaking training as part of the formal program, which was a really great, I think, aspect of this program. And many of the faculty mentors commented afterwards about what a great job these young investigators did, not only in the projects themselves, but also in presenting them. I thought we really could see the impact of that. And that's something that, is such an important skill for young investigators that they will bring forward at every meeting they go to, every presentation they do. So it was great. It was a great aspect of this.
And it builds confidence. That's
absolutely. Sometimes I think, especially in the post-COVID world, so many talks are virtual. I think actually having them get up in front of a group and stand at a podium and present their slides and sort of talk about how to put together a good PowerPoint. These things are incredibly important to conveying our research in a way that is, effective and efficient. So it was great to see. So, we had a number of different types of projects and abstracts presented, everything from ongoing clinical trials to basic science to case reports. So we'll sort of go through them and chat about them one by one. Starting to go through the specific abstracts that were presented, we kicked things off with Dr. Chinmay Jani from University of Miami, who presented an interesting study looking at the epidemiology of lung cancer, particularly with an eye on the young adult population, looking at incidence as well as risk factors. And, I think his work really highlighted the fact that while, lung cancer incidence overall is declining, it still remains a big problem among young adults. And that our understanding of risk factors associated with lung cancer in general, and particularly lung cancers that are arising in younger patients, those without classic lung cancer risk factors, remains inadequate, and that we need to do more to really understand why these cancers are developing. Next, we had Dr. Lani or Nora Chen from Columbia University, who I thought presented a really thought-provoking study looking at cachexia, which is sort of this term for weight loss and sort of decreased muscle mass in patients with EGFR mutant cancers treated with osimertinib. And she showed, which we've seen in other studies as well, that overall weight loss is very common in patients with
lung cancer and EGFR mutant cancers in particular. And that weight loss in and of itself is associated with inferior outcomes. But I think we have to understand that this process of weight loss and cachexia is really complex. And one of the things we talked a lot about in the Q&A is that we really don't understand sort of all the contributors to why this is so important, or importantly, how to modify it. What we can do, what we can guide patients as clinicians, and what patients can do to help mitigate this potential risk factor.
Yeah, I found this really interesting as well, that in her study, nearly half of the patients receiving, first line just targeted therapy met that criteria of losing greater than 5% of their weight in 6 months. And I agree with you. We really need to understand, is it the weight loss? Is it disease related? Is it drug related? Because there isn't one reason, I don't think, why people lose weight. And if everybody does lose weight, how severe is it? And does that impact outcomes as well? Is it reversible? There are so many questions, but it is so important, such important research. And hopefully there'll be a lot more that will, happen from this.
Yeah, I think every faculty member that, during that presentation said it's one of the things that comes up most commonly in a clinic visit. What should I do about my diet? How can I, what should I be eating? These are such important things that patients are thinking about and families are thinking about. So hopefully, a lot more to come and a better understanding of how we can modify that risk factor.
Yeah. And as someone who did lose weight, on a lot of weight on a TKI too, it's a lot easier to say, oh, you need to put on weight. It's a lot harder than it is, you think it would be, because you always say, people are always like, oh, it's so hard to lose weight. Put on weight. I could eat and eat and eat. It's just as hard to put on that weight. So it really, and it's hard to eat stomach stuff that, it's very complex. So yeah.
Absolutely. Yeah. So hopefully a lot more work to come out of this, in this group. I think a really important area to keep an eye on. Up next, we had Dr. Bilal Ode, who is actually a general surgery resident from UT Southwestern. This is pretty amazing. He rearranged his call schedule to be able to come up to the summit, and then was flying back to round on patients the very next day. So, incredible what some of these, particularly residents and fellows, are doing to be able to join us. And he presented, I think, also really interesting and sobering work looking at testing practices, looking at the rates of molecular testing. And once again, as many studies have shown, and again, emphasizing that we're not doing as well as we should be in terms of molecular testing, that there's a lot of variability by region and by hospital. And so, again, sobering work that is a good reminder that we still need, we can talk about targeted therapies all the time, but we need to ensure that all patients are being tested in order to know who might benefit from these different targeted therapies.
Mm-hmm. Yeah.
Then we had Dr. Teja Vorganti from University of Pennsylvania. He presented sort of a trial in progress, a study that they're doing at Penn looking at patient patterns and outcomes of lung cancer of young onset lung cancer. They're calling this the Spark Lung Study. And this is an interesting retrospective study looking at some large data sets, again, trying to understand young onset lung cancer. I think this has been sort of a theme of trying to understand this particular patient population, demographics, risk factors, as well as treatment outcomes. So this is sort of a project in progress, and we look forward to seeing more from Dr. Vorganti and the group at Penn. Up next, we had Dr. Barlise Weissengren, who is at Cedars-Sinai Medical Center in Los Angeles. And Dr. Weissengren presented a really interesting study, which essentially, was a retrospective study that started at Cedars-Sinai, but actually ultimately was expanded to also include Henry Ford Hospital in Detroit. And they looked retrospectively at patients in their centers with EGFR mutated lung cancer and outcomes, looking specifically by race. And I think this was really a sobering moment in the summit, because they clearly demonstrated in these two different institutions that there are inferior outcomes for patients with EGFR mutant lung cancer, looking at both progression-free but also overall survival when you look at Black or African American patients compared to other patient groups. And this was really striking to see. I think, we all our jaws sort of dropped looking at this. And again, it's not necessarily that it's new, but I think to see it here was really just so impactful. And there was a lot of discussion afterwards about why this might be. Is it, differences in how therapies are behaving in these patients? Is there sort of pharmacogenomic differences? Is there differences in other factors, for these patients that are impacting outcomes? And I think Dr. Weissengren and the groups at Cedars-Sinai and Henry Ford are, continuing to study this and really to try to understand what are the modifiable factors here? How can we do better for this patient group? But I thought a really important abstract.
I did too. And I was horrified. It was. And again, race plays a factor. We know that in general, but, there, when you look at the population, I think Cedars-Sinai was like 7 or 8% were Black. And when you look at Henry Ford, it's like 75%.
Yeah.
So you're looking at a population that's treated at a great hospital as well. So what are the other factors that contribute to this? And I like the idea. I do think that there are biologically distinct, things about us people. We know that in, certain cultures or ethnicities are more susceptible to certain, genetic diseases or mutations. And so I do think it really is important. And I look forward to them moving forward with more research, because it really could help guide treatment strategies for, different populations, and hopefully help, increase and improve survival.
Absolutely. And I think one of the neat things about it was that this also sparked so much discussion among participants from all different institutions. So my hope is that, this is something that maybe could even be expanded as a result of the summit to include other institutions to improve the power and understand this at even a broader level. So hopefully stay tuned for more. We really look forward to more work for Dr. Weissengren. Up next, we had a local hero, Dr. Robert Cameron, who was at University of Chicago, so didn't have far to travel to the summit. And Dr. Cameron presented a study looking at again, another trial in progress, looking at a study that's doing a novel method of isotope labeling to identify metabolic features of cancers that have become resistant to osimertinib. So this is a study where they're using kind of a novel technology in patients who are undergoing biopsies at the time of progression to try to understand how the metabolism of these cancer cells impacts resistance, and whether in the future this might be sort of a novel way to target and overcome resistance. So again, early days for this project, but I thought really an interesting and new approach. And again, I think it was neat to see this. We saw it in a few different studies, really sort of novel approaches, different ways of treating cancer that are, kind of pushing the envelope of what we've been doing until now. So great to see. Following that, we had Dr. Francisco Exposito, who is a PhD. He's a postdoctoral fellow at Yale University in Katie Politi's lab. And he kicked off a session that was really focused on more sort of preclinical laboratory-based work. His project in particular was focused on the role of co-mutations in EGFR-driven cancers, and specifically on a couple of tumor suppressor genes, STK11, also called LKB1, and SETD2, and how these might interact with EGFR mutant lung cancers. And I think the goal of this project is really to identify new therapeutic vulnerabilities, again, starting in preclinical models, starting in the lab, but ultimately hoping to translate that into potentially future clinical trials and new treatment options for patients. So I thought this was a great project as well.
I did too. And I really, I think sometimes we get so caught up in translational and clinical science, we forget that the basic science is, kind of the information that we can gain and how important it is. But I really, I thought it was interesting because we don't always know the implications of co-mutations, and especially tumor suppressor genes that, they really play more of an active role in tumor initiation and how the cancer can behave over time. So while I didn't understand it all, I found it very intriguing, and I asked him for a 101 if he could to explain it to me further, which he said he would do. But all in all, I just, I think, finding, going back to basic sometimes is what's going to move us, forward faster, more than some incremental advancement.
And I think a great thing about the summit is both in terms of the representation of the mentors and the participants, we try to always have a good mix of people who are more preclinically focused, translational investigators who are sort of bridging that gap between the lab and the clinic, and then more clinical investigators, because I think, it can be an opportunity to provide sort of feedback on how to best translate this into patients and clinical care. We also get great feedback from some of the mentors who have more lab-based backgrounds and who can really, dive into the details of the experimental design and help provide guidance there. So it's always, I think these discussions are always fascinating in these proposals or in these projects. So it was great to see. Next, we had Dr. Robert Su from University of Southern California. He's actually a junior faculty member, and he showed an interesting study looking at some of the sort of co-alterations or different kind of protein expression that can be seen in EGFR mutant lung cancers, specifically focused on estrogen receptor expression and association with outcomes in EGFR mutant lung cancer. So this was another one of these studies. We're really sort of trying to hone a little bit more in on what are some of the differences between patients who may have better outcomes versus worse outcomes on TKIs? Can we try to understand the biologic differences between those? And ultimately, in the long term, might these identify sort of opportunities for new combinations or new ways of targeting cancers that might help, patients do better than they might otherwise do with treatment? Up next, we had Dr. Benjamin Morris, another postdoctoral research fellow, in this case from the HAMAC lab at MD Anderson. And he presented, I thought, a really, beautifully presented presentation, but also a really interesting project looking at this population of cells we call the drug-tolerant persister cells, or DTPCs. And this idea of these drug-tolerant persister cells is a really important one, we think, in lung cancer or in cancer biology in general. What that refers to is patients who are ongoing kind of treatment and are on a treatment, say, for example, an EGFR inhibitor, and their cancer is responding. It's really asking, what are the cells that are surviving on that treatment, that are sort of hibernating and inactive, but ultimately are the seeds of resistance and how it develops. And there's always this concept that if we can target drug-tolerant persister cells in some way, we might be able to delay or prevent the emergence of resistance. And so Dr. Morris and his group in Dr. Hamac's lab did a sort of in-depth analysis of the kind of gene expression of these DTPCs and identified KRT17 as a particular gene upregulated in DTPCs that seems to be sort of an important mediator of, sort of pluripotent cells that have the potential to develop into various resistance mechanisms. And so I am probably not doing justice to all the biologic details of the work that they did, but the idea here is that, we're understanding the biology of these cells so that in the future we may be able to target a population of cells like this with another therapy and potentially improve long-term outcomes for patients. So again, this one was one that generated a lot of discussion and a lot of excitement.
Yeah, and it is. And it's really, I think, the drug tolerant persister cells are a major challenge, right? And, what I got out of this presentation too was that these KRT17 cells emerge at that state, at the drug tolerant persister, cell state, as opposed to resistance, right? Mm-hmm. So I thought that was really important and interesting. I did have a discussion with him after about, how would you target it? Because this, KRT or keratin 17 also our healthy tissues use it, right? To repair, wounds or whatever. And so we kind of had a little discussion about that, and he had some ideas. He was really excited, but this is the first step in that work. And I think I look forward to seeing more of work in this area. He is really passionate about So I have no doubt we will see more from him.
Absolutely. Absolutely. I think, again, this idea of studying the persister cell biology is so important, I think, to moving the field forward. So great to see.
Mm-hmm.
And then I think for the last section of the afternoon, we moved back into sort of a more clinically oriented group of abstracts, starting off with Dr. Muskan Agarwal, who's an oncology fellow at MedStar Georgetown Hospital. She presented a really interesting case report of a patient that they had cared for who had a lung cancer with an EGFR exon 19 deletion, but also a concurrent germline, so sort of inherited a mutation in BRCA2, which is, of course, a gene well known to be associated with risk for a variety of different cancers. And she really talked about sort of the treatment of this patient and how these germline BRCA2 mutations might impact, the treatment options that we think about, knowing that sometimes BRCA2 mutant cancers have particular vulnerability to chemotherapies, as well as some other drugs like PARP inhibitors. And so this was, sort of an individual case report, but I think raised a lot of important issues that are relevant to many patients.
Yeah. Yeah. This was really interesting. And there were, when she was giving the presentation, a couple of things, first of all, we're constantly telling people when you have biomarker testing, they're not looking at germline, they're not looking at your DNA, they're looking at the cancer. So, here they're looking at both the cancer and your DNA. So, what does it matter, right? What role does it play if it's germline? If it's germline, is it always in the cancer? What are the implications? There are so many questions. And as of now, most people don't go for germline testing. We don't get genetic counseling, if you will, for the most part, unless, something looks like it is really, may be in your germline. So I think there needs to be a lot more work in this area. So I really appreciated her bringing that up and starting to do the research so that we do understand that better.
Yeah, it's really interesting. There's actually some of the molecular testing companies are actually starting to incorporate germline testing. It's really when we talk about sort of cancer DNA or somatic mutations, which are mutations that are only present in cancer cells but not in normal tissues, that's in contrast to germline, which are mutations that are present basically in every cell in a patient's body and therefore are, something that's passed along in families. But in order to test for that, you can either look at the sort of frequency, the prevalence of that mutation in a tumor, or, more commonly, what you can do in a blood sample is actually test normal cells. So healthy white blood cells, for example, or other sort of normal cells and look for mutations there. So there are opportunities to do this testing. And you're right. I don't think we're probably doing quite enough of it. I don't think we fully understand, exactly when it should be and shouldn't be done. Of course, it has lots of implications for patients and families. But I think this is starting to become something that's being talked about more and more, both in a research perspective and in clinical care.
Yeah. And thankfully, genetic counseling is a thing because
Absolutely.
it's not when you're necessarily talking about biomarker testing. And just an example, I have a germline LATS1, LATS1 that showed up in tissue, my last biopsy. Nobody has any idea what the implication. I'm okay with that, right? I can understand that it may mean something, it may not. But to just give somebody who, is not involved in any of this, that kind of information, what do they do with that information? Yeah. So I do think, I think it's important, but I think it should only be done with genetic counseling.
Yeah. Because it has such, I think what a patient, what a family does with that information is so important. And so I think, if you as patients, have questions about this, talking to your clinicians, asking about genetic counseling is never the wrong thing to do.
Right.
So then we had Dr. Vishwanathan Neal, who is an oncology fellow at Henry Ford Hospital. He presented a case series on a topic that we've really seen, almost every year, I think every year at the summit, which is histologic transformations in lung cancer. He presented, a case series of transformed EGFR mutant, initially non-small cell lung cancer, transforming to small cell lung cancer. I think this is always an important topic to talk about. This can happen in patients early who don't really have the type of response that we're hoping to see to an EGFR inhibitor. Of course, it can also happen later at the time of what we call acquired resistance after a time of response. And, I think, again, it really just highlighted that this is a recurrent issue that can come up, that it can currently only be detected by tissue biopsies at the time of progression. It can't be detected by liquid biopsies. And once again, emphasize that there's a lot more work still to be done to understand histologic transformations. Up next, we had Dr. Jackie Oredo, who I think gets the record for the most summits attended. This was actually her third EGFR resistor summit. She started, I think, as a medical student the first time she came, which is really pretty incredible to watch, such a rising star in lung cancer from medical school through fellowship, through residency, and now to fellowship. And so she's now at Stanford. She's an oncology fellow at Stanford, and she presented sort of a trials in progress, but a trial that's nearly completed now, that started as an investigator-initiated trial and was such a sort of interesting concept that it became a industry-sponsored larger global trial, which is really cool to see. This was a study looking at patients with unresectable EGFR mutated lung cancer. Those patients currently are treated with concurrent chemotherapy and radiation, followed by osimertinib. We saw that was that became the standard of care based on the LAURA trial that was presented a couple of years ago. But here they're really looking at the role of induction osimertinib. So for patients like this, rather than jumping right into chemotherapy and radiation, might giving them a period of EGFR inhibitor therapy actually help improve their long-term outcomes, shrink tumors, and help sort of make a smaller radiation field. And equally importantly, is that a safe thing to do? How does that impact outcomes on, the radiation portion of the treatment? Thinking about things like, the risks of pneumonitis or lung inflammation. So what Jackie presented was really a trials in progress, sort of, reviewing the data that has led to this study and the study design. But it was actually exciting. She had to be very careful about what she said because they're going to be presenting some early results from that study, I think, soon at the ESMO Asia meeting. So we really look forward to seeing more data from this Neola trial, but also looking forward to see what Jackie, does next, because she's such a fantastic young investigator.
Yeah, it's been great to see her grow over the years. I really, really enjoyed it. And she was really good at skirting the question that everybody was asking.
Like a pro.
Like a pro. But, again, from the patient perspective, what I like about this is, I think first of all, you would get to see a response. How is it going to respond to the osimertinib, right?
Mm-hmm.
Think that is important to see and gives you more faith when you go on it as maintenance. But the other is debulking it. Yeah, like your lung is finite, right? If you can, only radiate half as much lung tissue, I think that's huge. Again, safety, we have to make sure it's safe. We have to, there are a lot of questions, but ultimately, if it can improve outcomes and quality of life, then definitely, I'm excited to see that data soon.
Yeah. And this comes up in clinic all the time of how to approach these patients. Right. So it's really important. And then the last couple abstracts, we had Dr. Christopher Grant, who's a oncology fellow at University of California in San Diego. He presented a really interesting case report of a patient who was put on osimertinib and developed a rare sort of dermatologic a toxicity that was thought to be sort of an allergic reaction to the therapy. And they worked together with the allergy team at their hospital to develop this lengthened desensitization protocol. So the idea being sort of to expose the patient to osimertinib in very small doses initially and slowly over weeks increase the dose to desensitize them to that reaction. And they were able to successfully do this and treat the patient. So again, a single case report, but a really interesting and intriguing one that could have implications for other patients.
We do it with antibiotics. Can we do it with, cancer treatments? If it's feasible, it would allow more people to go back on effective treatments as opposed to having to start a brand new treatment. So it's interesting. It was innovative, definitely.
Absolutely. Absolutely. And then we had, last but not least, Dr. Malcolm Sue, a resident at UT Southwestern. He presented a retrospective study looking at central nervous system or brain and leptomeningeal metastases, both the sort of incidence on treatment and then patterns of progression after patients had either continued or discontinued osimertinib. So this was a small study, but I think it really highlighted something that many of us think a lot about, which is, how does this, for example, stopping osimertinib at the time of progression impact the rate of development or progression of CNS metastases? Because we know that osimertinib is a very CNS penetrant therapy. And so again, this was, really intriguing. I think it has implications for us as we think about sort of next line treatment options for our patients. How do we incorporate both sort of novel therapies like amivantamab and others, but where do we see the role of TKIs in that patient's care? So, I think more to come on these questions, but really important ones in the clinic. So then, I think we just wanted to also highlight that throughout this day, as Jill already mentioned, we had sort of many kind of small group mentoring sessions. We try to rotate the young investigators so they got, time with both Jill and the patient advocates and Chris Kimball, as well as with all of the different faculty members. And then we also brought in two, I thought, really wonderful presentations throughout the day. The first was Jill herself, who gave a wonderful talk about how we can work together, how the research community, the oncology community can work most effectively together with patient advocates. Jill, I thought your talk was spot on and really terrific. And I thought it was such an important message for these young investigators, because this is something that is a part of being a oncologist or a researcher in lung cancer these days. It's something that brings richness and brings impact to the work that we do. And it's also something that's mandatory for many of the grants we're applying for in research projects. And so, I think knowing how to do that, how to do it respectfully and effectively, and how to maximize those interactions is so important. So I thought you did a great job highlighting that.
Thank you. Thank you. Yeah. And I think it was important too, because, as I told them, you don't learn it in school. Your degree doesn't depend on it. It's not part of fellowship. There are time constraints. Yet, as you said, they're required. So whatever we can do to put them in a position to be successful early on in their career, I think just benefits everyone.
Absolutely. And it's, one of the great things I think about having them participate in programs like this so early in their career, right? They become sort of something that they learn early on and they can use throughout their whole career. And then we had Dr. Haymack, who gave a great talk, as he has in past sessions as well, talking about NIH funding and grant writing. And really, I think sort of taking a 10,000-foot view at how you think about your longitudinal career, starting from a young investigator and sort of how careers evolve, thinking about which grants to apply for, how to think about projects. I thought it was a really wonderful talk and, really struck the right chord, I thought, both for the basic science researchers, but also for the clinical investigators. So he was really wonderful, and we were grateful for him for doing that. Yeah. So the day wrapped up, at the end with a closing reception, and every year the faculty mentors together with Jill and the EGFR resistors, we sort of have to make the difficult choices to pick the top abstracts. Even though there are many wonderful abstracts, we actually the judges do go through and score each of them. We score them based on impact, based on sort of novelty, as well as presentation. All of those, how these young investigators present their work as well. And so each year we try to pick the top abstract. So this year we awarded three awards. The second runner-up was Dr. Barlise Weissengren, who had that project looking at reduced survival in Black patients with EGFR mutant lung cancer. Again, this was really one that I think struck a chord with everyone, and we thought was a highly deserving of an award, and really, work that will deserve a lot more attention going forward. And then our first runner-up was Dr. Jackie Aredo with the Neola study. This was the study of induction osimertinib prior to chemoradiotherapy and then maintenance osimertinib. She, I think, won both because of her excellent presentation, but because this is such an important and clinically relevant, trial concept. And I think all of us are really excited to see those results come forward. And I think they have the potential to really impact clinical practice. Mm-hmm. And then, the grand prize, which last year we renamed the EGFR Resistors Ivy Elkins Award in honor of Ivy and to her incredible contributions to really founding this whole summit, and to her deep commitment to, the next generation of researchers and supporting them and working together with them. I think it's really, wonderful that this is now the Ivy Elkins Award. And this year it went to Dr. Benjamin Morris from MD Anderson, who had that work looking at the drug-tolerant persistent cells and the role of keratin 17 in that cell population. So again, a great presentation and a really thought-provoking and project that has the potential to really have an impact on patients with lung cancer. So, all of the presentations were wonderful. All of the research was great, but these three won the top awards, and I think were well deserved.
Yes, I agree. I think, and it is always hard because everybody has a wonderful project and they're all important. So it is always hard, I think. But I think everybody understands how much we appreciate them anyway, and how wonderful it is to have them a part of the summit. But so thank you, Dr. Piotrowska. That was a great, perfect summary, as always. You explain it so well. And, once again, that's why I said the future is bright. They are the future. I'm really excited about that. I think, I want to just reiterate that the summit does have a meaningful impact on their professional development, and they are the next generation of lung cancer researchers. But, that's really important right now because there's a lot of funding that's being cut. So I think as the EGFR resistors, it's even more important for us to really support these early career investigators to keep them in the space. So that was really important. And I think, that the summit itself, again, that intimate setting, they don't get this experience anywhere else. And, it is hard to explain, but it is truly inspiring. One of my most favorite advocacy work that I do, and it fills my heart with hope and gratitude, a lot of gratitude. But I want to also just recognize the unwavering support from our mentor, judges, faculty, because they take time out of their busy schedule. And there's so much going on right now and so many conferences, and what they did, some of them, to be sure that they were here was amazing. It was that important to them to create this kind of space for early career investigators to learn and to grow. They shared more than just their time and their expertise. They really shared themselves, and it's wonderful to do that. But also, so thank you.
I think just to say, it's really a highlight of the year for many of us too. I think we leave inspired, right? I think we leave inspired by the young investigators. Every year we also leave inspired by you, Jill, and by the EGFR resistors and all that you guys have accomplished. So, it's such a wonderful couple of days. I think every year it's so invigorating in a busy time.
Yeah, I'm glad that you guys feel the same too. Sometimes you need that, right?
Absolutely.
Like, it's that good for the soul to go
Absolutely.
back to the true north of everything.
That's right. I
think that's, it's crazy. It's wonderful. So wonderful. But, so, I also want to do a couple more thank yous. The summit is truly only possible because of the incredible partnership with CC, your tireless efforts, your patience, creativity, the energy, and, just the support you put behind the scenes to help us is nothing short of amazing. And thank you from the bottom of our hearts. We are also deeply grateful for the generous continued grant funding. It's long-standing support from AstraZeneca. Your partnership over the past 7 years has allowed us to build and sustain this important event, this important summit. And we are so grateful. And I think I could speak for everybody. We look forward to many more years of partnering. And on behalf of the entire EGFR resistors, patient and caregiver community, thank you. Thank you to everyone. The work you're doing is helping people with EGFR positive lung cancer live longer and live better. And we are very grateful for all of your support. Thank you.
Thanks, everyone.